right, welcome back to the afternoon. So we have two more fellow presentations. The first presentation is going to be by Kishor Pant, who's a researcher in Dr. Sergio Grattaloni's Laboratory of Cancer Cell Biology and Translation Research at the Hormel Institute, University of Minnesota. It also says Austin, USA. Dr. Kishor is mainly engaged in the research of hepatocellular carcinoma, cholangiocarcinoma, and polycystic liver disease. Currently, he works on primary cilia-mediated regulation of the cell growth and cellular metabolism. And that is what he's going to present to us, the role of primary cilia and EGFR signaling regulation in cholangiocarcinoma. Dr. Pant. This is forward and this is your pointer.
Thank you. And my name is Kishor, it's already introduced, and today I'm going to talk about our work in progress and the role of primary cilia in the EGFR signaling regulation in cholangiocarcinoma. Oh, okay. So cilia are an organelle found in almost all cells in the human body. They are microtubule-based organelle, and they are basically two kinds. And one is called motile cilia, which has the main function to move the extracellular fluid around the cells or the tissue. And the other is basically primary cilia. It's also considered as a sensory cilia, which is found in the liver, skin, and other cells and other tissue. Which is okay, and our main focus is to study the role of primary cilia and its function in the cell growth and metabolism and other function in especially in cholangiocytes. So cilia is primary cilia also considered as a cellular antenna, which found in the apical membrane of the cells, which extended out of the cell membrane. And cilia, primary cilia, which sends the mechanical, chemical, osmotic signals, and these signals are transduced further into the cells and which affect the cellular functions. So as I already mentioned, cholangiocytes, they also have primary cilia. So bile duct, if we zoom into this electron micrographs, you can see the cholangiocyte, the lining of the bile ducts. So they have cilia in their lumen, and which has several functions in the liver. So primary cilia, and what we have found that primary cilia is found to be either defective or lost in the cholangiocarcinoma as well as polycystic liver disease. And in our previous study, we have shown that in cholangiocarcinoma patients, the primary cilia is lost from the lumen of the bile duct. In same way, we also saw the in vitro samples also show that in normal cell there is a cilia, however, the cholangiocarcinoma cells, they lost their cilia. And in polycystic liver disease also, we saw that there is a report, they have shown that the cilia is lost or defective in the cystic liver disease cells. So what is common in these two cells? They both have increased cell growth as well as the increased EGFR signaling in these cells. So there is also several reports that there is increased EGFR signaling in both in cholangiocarcinoma as well as polycystic liver disease, and this can be a potential therapeutic target in the cholangiocarcinoma as well as the polycystic liver disease. So our focus is to study the role of cilia. Then we decided to study the role of mechanism and underlying the increased EGFR activity in cholangiocytes with the cilia defects. And we hypothesized that primary cilia can regulate EGFR signaling. So for this, first we prepared our animal models. We knock out IFT88 ciliary proteins from these animals, and we can see the cilia stain. And wild type animal, there is a cilia in the lumen. However, in knockout animals, there is a cilia is lost. We also use in vitro model. We use normal human cholangiocyte NSC69. We also deleted ciliary protein to make ciliary defective cells. And we use HU61 cell cancer CCA cell line as well as to compare our results. Same way we use normal rat cholangiocyte NRC and the polycystic rat cholangiocytes. So first we check what's happened after we removed cilia from the cholangiocyte, and we can see that in the IFTT cilia knockdown cells, there is a increase in enrichment of the EGFR signaling proteins. And when we check the protein expression, when there is increased EGFR signaling, either with the genetic knockout method or with the chemical desensitization method with the chlorhydrate. So which indicate that cilia defects can induce EGFR signaling in cholangiocytes. Next, we check the EGFR signaling, increased EGFR. So we found that the cilia defective cholangiocytes in these animals, there was an increased EGFR activity as well as the MAP kinase in these animals. So as we already know, as soon as EGFR is bind to its ligand, it's activated at this cytosolic side with the phosphorylation. Then it's recognized by the CBL protein. Further is lead to the endocytosis and the ubiquitin, which leads to the degradation of the EGFR. This is in the normal process. This thing required for the EGFR clearance. And what happened when we treated our cells, cilia defective cells with the EGF in the time 1 minute to 2 hours? So we found that there is a increased sensitivity of the cilia defective cholangiocyte compared to normal cholangiocyte. In same way, we found that after 1 hour of treatment, the cilia defective cholangiocyte, they have increased EGF sensitivity and as well as the cholangiocarcinoma cell lines. Similarly, when we inhibited EGFR activity with the AG1478, the EGFR activity was decreased as expected. So we asked question why EGFR is more active in the cilia defective cholangiocyte. So for this, we removed isolated cilia from the normal rat cholangiocyte as well as the polycystic liver disease cholangiocytes, and we used those cilia protein for the proteomics analysis. And we found that the normal rat cholangiocyte, NSC, they have increased EGFR in their cilia compared to polycystic liver disease cholangiocyte. And unfortunately, we cannot do this for the cholangiocarcinoma cell because they don't have cilia in their cell surface. So we also repeated this in the immunoprecipitation analysis. We see this EGFR which interact with the ciliary protein. So it is confirmed that EGFR can interact with ciliary proteins. And next we asked, so what could be the main reason? Like next we went to the check the endocytosis. We found the dinosaur which inhibit the mainly endocytosis process. We found in normal cholangiocyte that dinosaur which inhibit endocytosis, which restore the active EGFR after EGF treatments. But in cilia defective cholangiocyte, it has a little effect. So it indicates that this cilia defect may induce some delayed EGFR endocytosis. Next, we also use, after endocytosis, we also wanted to check the clearance of the EGFR from the cells. We use 2 proteasome degrader inhibitor. So we use these 2 for our experiment, and we found that in normal rat cholangiocytes and human cholangiocyte, there is an increased EGFR, active EGFR after treatment of the proteasome degrader compared to the, but there was a little effect in the cilia defective cholangiocytes. And then we asked what happened when we restore primary cilia in the cholangiocyte, what will happen to EGFR activity. So there is this in our lab we have been doing using HDAC6 inhibitor, which known to induce EGFR, which known to induce cilia restoration in cholangiocarcinoma cells. So we use either HDAC6 shRNA, tuberstatin A, ACY. These are the 3 different methods we use for the HDAC6 inhibition. And we found that these inhibition of HDAC6 induce cilia in cholangiocarcinoma cells. However, when we treated these cells, in normal untreated cells, the EGFR activity was sustained even after 2 hours and 3 hours in the cholangiocarcinoma cells. However, when we treated with the HDAC6 inhibitor, either ACY or the HDAC6, the EGFR activity decreased significantly in these cells. Similarly, we repeated this experiment in the rat cholangiocarcinoma cell BDNU, where we can see the tuberstatin A, another HDAC6 inhibitor, which decreased the EGFR activity in these cholangiocytes. Next, when we use these BDNU cells to prepare orthotropic rat animals, where we treated these animals with the tuberstatin A. We found these tuberstatin A induce cilia in these tumors as well as decrease the EGFR. And these EGFR as well as the cilia localized with EGFR. So, which indicate that the HDAC6 inhibition can lead to the EGFR clearance and the cilia formation. So we also found that these treatment of tuberstatin A induce inhibited the phospho-ERK and phosphomapanic activity in these animals' tumor as well. And so in summary, we observed that cilia defect can promote dysregulated EGFR signaling in cholangiocytes, and defects in primary cilia which lead to the disruption of the activated EGFR clearance. And HDAC6 inhibition can induce primary cilia restoration as well as suppress the EGFR signaling in cholangiocarcinoma. In future study, we are looking at the role of EGFR regulator like CBL interaction with cilia, how they can regulate cilia and EGFR clearance. We are also which I already mentioned, HDAC6 linked with the cilia in EGFR clearance, as well as we are also now focusing on cilia-mediated signaling in EGFR and FGFR drug-resistant cell lines to check on ourselves. And with this, I would like to thank my team and my PI Sergio Gratilonen and our collaborators. Thank you very much.
Great. And our next fellow presentation is going to be from Dr. Vindhya Vijay. Dr. Vijay is a postdoctoral fellow at Massachusetts General Hospital Cancer Center and Harvard Medical School. Her current work is focused on developing and characterizing patient-derived and genetically engineered mouse models to study various genetic subsets of cholangiocarcinoma with a focus on BAP1 mutant cholangiocarcinoma.
Thank you so much for this opportunity. Today I'm going to be talking about a subset of cholangiocarcinoma that's driven by the loss-of-function mutations of a gene called BAP1. So BAP1, or BRCA-associated protein 1, is mutated in about 20% of all ICCs. We normally see truncating mutations or deep deletions, which leads to ultimately leads to the loss of the protein BAP1. And when you look at BAP1, it is a deubiquitinating enzyme that belongs to the ubiquitin carboxyhydrolase family. Several functions have been associated with BAP1, the major one being BAP1 and its core complex can deubiquitinate histone H2, which regulates transcription of a majority of genes that are involved in cell cycle differentiation and metabolism. BAP1 is also known to regulate substrate stability, for example, in DNA repair factors as well as other cytoplasmic substrates, and can also directly regulate transcription when they're recruited directly by the transcription factors. And this is a mechanism that's independent of its deubiquitinase activity. So if you look at BAP1 mutant patients, there's a subset of these patients that have co-occurring actionable mutations in IDH, FGFR2, and BRAF. But there's this remaining set of patients that don't have any actionable mutations, which makes it really important for us to identify alternate strategies to target this subset of patients. So really going to this, we have two key questions. What is the oncogenic program that's unleashed by BAP1 loss within the liver? And by understanding this, can we identify molecular dependencies that we can take advantage of for treating BAP1 mutant ICCs? So a key challenge has been clinically relevant models. So we developed a genetically engineered mouse model by using a liver-specific albumin promoter to knock out BAP1. And we found that homozygous deletion of BAP1 resulted in significantly decreased tumor-free survival and also ICC, as shown here in histology. And when we acutely deleted BAP1 in adult livers, we found that there was significant increase in the liver size and also rapid cell cycle entry, as shown here with the Ki67 staining. So these results went on to show that BAP1 plays an essential role in maintaining quiescence within the liver. When we profiled these livers, what we found was that upon BAP1 loss, there was a significant increase in the lipid metabolism program, especially those genes that are involved in sterol, cholesterol, and lipid biosynthesis. The heat map here on the right is showing you the leading edge genes that showed the lipogenic signature. But importantly, when we looked at the patient data set, we found that at both the mRNA as well as the protein level, there was a significant increase in the lipogenic signature or enhancement of a lipogenic program in BAP1 mutant ICCs in comparison to other genetic subsets such as IDH, KRAS, as well as P53. In the last couple of days, we've talked about the importance of good models. So a huge ongoing effort in our lab has been to develop preclinical models or patient-derived cell line models that we've subjected to genomic, transcriptomic, as well as proteomic profiling. And what we've also done is functional genomics to create this atlas of biliary tract cancers where we can identify specific vulnerabilities that are associated with different subsets of BTCs. So when we analyzed these models, it further reinforced the finding that the lipid metabolism is a significant feature of BAP1 mutant ICCs in comparison to other ICCs. So we all know metabolic reprogramming is very important in cancer. We've seen that these mechanisms are constantly altered by different oncogenes and tumor suppressors to handle the nutrient stress. One of the questions that we had was that these molecular programs or molecular circuitry that's altered upon BAP1 loss, do these mechanisms are these mechanisms required for continued, or do they have a continued role in established ICCs? And if so, do these mechanisms directly regulate lipogenesis? So we used a BAP1 deficient model, and we restored wild-type or fully functional BAP1, or a catalytically dead or a mutant version, which is a C91A BAP1. And we found that upon BAP1 restoration, there was significant decrease of cell proliferation. And this was specific to the BAP1 deficient ICCs, but not in BAP1 wild-type setting. And we also found this to be true in vivo, where BAP1 restoration resulted in significantly smaller tumors or no tumors at all. So these results go on to show that BAP1 has a continued role in maintaining ICC tumorigenesis. Since our profiling showed lipogenesis as a prominent feature of BAP1 mutant ICCs, we stained our cells using oil red O, which is a stain to visualize intracellular lipid accumulation. And we found BAP1 deficient ICCs, in comparison to wild-type ICCs, had significantly higher levels of lipid accumulation within the cell. And when we restored BAP1 levels, we found that restoring BAP1 levels was able to decrease these levels of intracellular lipid accumulation. And in contrast, when we deleted BAP1 in the wild-type setting, we were able to see an increase in the intracellular lipid accumulation. So these results show that BAP1 does play a role in functionally regulating lipogenesis. So we wanted to see what are the other what is the mechanism by which BAP1 was regulating lipogenesis, and we found that the transcription factor that was most highly deregulated upon BAP1 loss was SRBP1, which is a lipid metabolism transcription factor, and it's a master regulator of lipid metabolism. In fact, we found that the transcriptional program of SRBP was significantly suppressed upon BAP1 restoration, and we've seen that in our human cell line systems as well. And we also saw that the mRNA levels of SRBP1 and SRBP2 itself was decreased upon BAP1 restoration. Interestingly, when we looked at our functional genomic screen data, we found that BAP1 mutant ICCs were selectively dependent on SRBP. In fact, it was the top dependency that we had seen. And it was not only SRBP, but we also saw other members of its pathway, such as SED, which is a transcriptional target of SRBP1. And we also saw SCAP and MBTPS1 that are factors known to activate SRBP. So we knocked out SRBP1 in a BAP1 deficient as well as wild-type model, and we found that deleting SRBP1 significantly decreased cell proliferation in BAP1 wild-type, in BAP1 mutant cell lines, but not in BAP1 wild-type cell lines. And true to what we were seeing with the lipogenic phenotype that was observed in BAP1 mutant settings, SRBP deletion was able to significantly decrease or reverse this phenotype. So normally when we have these high lipid overload, what we see is that increased oxidative stress, which results in a form of cell death that's called ferroptosis. It's an ion-mediated form of cell death. And there are compensatory mechanisms within the cell that can offset this, such as increased glutamate uptake and glutathione metabolism. So one of the questions we had was that with this high lipid overload, how are we not seeing cell death? So we found that BAP1 loss resulted in increased levels of anti-ferroptosis factors, such as GPX4 and ACSL3. But at the same time, we saw a decrease in the proferroptotic markers, such as ACL4 and NOX4. So these results went on to show that there was not only high lipid biosynthesis that was happening, but this was accompanied by a protective mechanism that caused resistance to ferroptosis. So we tested this by looking at lipid peroxidation. So we used a dye that turned from red to green upon lipid peroxidation. And you can see that BAP1 restoration was able to significantly increase the levels of lipid peroxidation based on the green signal there. And when we used a redox scavenger, such as N-acetylcysteine, we were able to reverse this effect with the increased red signal that you can see. We also looked at protein carbonylation, which is another sign of oxidative stress, and we once again found that BAP1 restoration was able to significantly increase protein carbonylation, which goes on to show that upon BAP1 loss, what we see is high lipid overload accompanied by decreased lipid peroxidation and carbonylation, which means decreased oxidative stress and ferroptosis. So in conclusion, what we've seen is that BAP1 loss, part of its oncogenic mechanism is to increase de novo lipid biosynthesis via SRBP1, which activates its transcriptional program. And restoring BAP1 levels was able to decrease this and not only decrease the lipid load, but also increase ferroptosis. Now, why is this important? Because this really opens up a scientific rationale for testing inhibitors of the SRBP1 pathway for BAP1 mutant ICCs, such as SCD1 inhibitors, FASN inhibitors, inhibitors of the mevalonate pathway for this subset of patients. With that, I'd like to acknowledge my lab, starting off with my mentor, Nabil Bardisi, and a shout out to Lei, Lilian, and Ines, who've contributed to this work, and our terrific team out there at MGH. We also have a lot of great collaborators at MGH and Broad. And finally, I'd like to thank CCF, the patients and families, for a couple of things. One, the fellowship that has really helped support my work over the last year, but also all of these studies wouldn't be possible if we didn't have the right models to ask these questions. So thank you so much.